Welcome everyone to the Lyme Disease Resource Center educational series. Our mission is to be a peer-guided, supportive, and safe place for those living with Lyme disease and their care partners, sharing strategies and resources for living well. The Lyme Disease Resource Center is an all-volunteer, community-based 501c3 organization, and were founded in 2013, celebrating 11 years. All of our services are offered completely free of charge. This evening's event, we have Paul Killinger from TickReport, and we're going to have a conversation after Paul presents about the latest in tick testing. Paul has directed education and outreach efforts for the TickReport testing service since 2018. Much of their research and service is focused close to home in Western Massachusetts. However, the lab regularly tests ticks from more than 20 countries across 6 continents. The lab's approach to passive surveillance of ticks and tick bites provides valuable insight on the seasonal activity of ticks, as well as the geographic distribution of tick species and pathogens. And I'd like to welcome once again Paul Killinger. Thank you so much, Paul, for joining us this evening.
Thank you very much. Hello, everyone. It's very nice to be here. I'm going to bring up a PowerPoint here. Give me just a second. We'll see how well it wants to cooperate with us. All right. Are we seeing the right one, or do I have to switch?
It's good.
Okay. Then I'm looking at a different screen than I'm accustomed to, but that's all right. Let me get the root of many troubles off the screen here, and just say a little bit about TickReport and what makes us a little bit different. So what TickReport primarily does is passive surveillance, and that means that everything is based on someone or a person or an animal close to them getting bitten by a tick, and they remove that tick, they send it to our lab for testing. The alternative, and what really drives a lot of, especially publications that make a lot of news, is active surveillance. And that's really where, instead of a particular bite, or instead of being so involved with sort of human activity, it's really land focused. So what will typically happen is you will have an assigned plot of land somewhere. In most cases, it will be out somewhere where maybe around here it would be in restricted parts of the Quabbin Reservoir, because frequently you're looking for places where people are not going through and messing with your experiment too much. And the active surveillance is not bad, because it's a good way to sort of control things, and you can say fairly accurately, year to year, changes that might happen in that particular plot. Usually we're talking a plot that's maybe 3 feet wide by 100, maybe a couple 100 feet long, and you would go and you would drag a piece of cloth across that plot, and then every several yards or something, you would be flipping that over, and there's this sort of mad rush by technicians with tweezers to pull all the ticks off of that flag as they can. We do a little bit of that, partly for training and education, and we have some videos on social media that get lots of reactions. But at TickReport, we're really looking at that passive surveillance, both because it has specific meaning for individuals who are bitten. So it's not just this this data point that's going to be put into a journal and read by, if we're lucky, several hundred people. This is something that has immediate relevance for the person who is bitten, and tells them something about the possibility of exposure to pathogens from that bite. And this map here shows, this is a couple years old now. I haven't bothered to update it because the map still makes the point. These are, every pin in that map is a unique zip code from which we've received a tick that bit a person or an animal. Definitely a strong sort of bias toward the eastern US, but there are ticks just about everywhere. There are different kinds of ticks in different areas. So this map, a lot of it really is, it's less a function of where the ticks are and aren't. There are more ticks in these areas, but in general, it tends to be more of a function of where the people are. If we compare this to a population density map, we would see an awful lot of people in those areas. And so with passive surveillance data, because we're just open to whatever is happening, if a tick is biting, we're not sort of predetermining anything. We learn a lot about the life cycle of these ticks. So this is this chart shows a single year of tick submissions to our lab, and each column is a single week. So we start out over on the left, that's just the first week in January, all the way to the right, first or last week in December. The top chart here shows the life stage of ticks that are coming in. So the green bars are adult ticks. We see that they're active first in the spring and are very busy, and as it gets hotter and drier, they tend to be less active, and so we'll see them fade. And in that process, the yellow bars are the nymph stage ticks. And so they are sort of the teenagers in the tick world. They have fed on one host and molted into the nymph stage, and now they're looking for a slightly larger animal to feed on. And they need a little bit more predictability in their weather, so they're not quite as tough in the spring, and just because of some other behaviors and animals being available a little bit later, they're a bit more active in the late spring and early summer. There are some little blue bars hidden in there, and those are larval stage ticks. We don't get quite as many of those, partly because they have a much lower infection rate, but humans don't encounter them quite as often as the nymph and adult stage ticks. So those are larvae that have just hatched from the egg. They're looking for their first victim, and they tend to run into rodents and other small animals the most. The way that humans tend to see them most often is with dogs, because dogs make poor decisions and and roll around in areas off trail, and they can pick up dozens or even a couple of hundred at a time. So larvae are unusual to encounter in solo. Just find one of them. Usually you're going to have zero or far too many attached to you, because they have this tiny little pile of eggs that's probably the size of my thumbnail maybe, and then those hatch and they spread out, and it could be a couple thousand of these larvae across an area just a couple feet wide, and they aren't traveling very far. They're just sort of spreading out, creating a little circle and waiting for an animal to stumble through. The lower chart here, those are different types of tick. So the blue bars are Ixodes, members of the Ixodes genus, most commonly the deer tick. The orange bars are dermacenters, like the American dog tick, and the green bars are amblyomma ticks, like the Lone Star tick. And so we see that there's definitely times that are of the year that are much busier and that there's a much higher risk of tick bites. And there are times of year when we are frankly kind of bored in the lab. And that's a time that we're working on other research and we're doing sort of recovery and data analysis from the previous year. But the fact that we're open all the time means that we're capturing these patterns and we're gathering this data that also lets us see if these patterns are changing over time. With the active surveillance model, where you go out and you're collecting ticks on a flag, I'm generalizing a little bit. This isn't the only thing that active surveillance does, but it tends to really focus on the summer months when you know that tick activity is going to be very high, and so you get a large sample size, which adds some significance to your results. And on a much more practical level, it's also when professors are not teaching, so they're able to go do field work for the summer, and students are not going to class. So there's frankly cheap labor available with students working through the summer. And it is very, very valuable. This is not necessarily one is better than the other, but passive surveillance is a way that we really think we've made a difference and done something that really adds something new to the conversation about ticks over the last 20 years, because we started this service open to the public in 2006. One of the questions that I always get is asking about how are tick infection rates changing over time. And especially when we hear about publications and news stories, there's a lot of talk about the rise of tick-borne illness and sort of new pathogens emerging. I tend to look at the news and social media and say, well, some of this is actually very positive because we don't see those rises in the ticks themselves. So in many cases, I think that those news stories are actually very positive because it's just a difference in awareness. Because people are actually, whether it's researchers or doctors or just individuals, they aren't they're just sort of blindly going out and potentially being exposed to tick bites that they're not worrying about. They are watching for ticks. They're looking to make sure that they're not exposed to the bacteria that causes Lyme disease. And there's a lot more awareness of all the other things that ticks carry. Lyme bacteria is certainly the biggest risk from a tick bite anywhere in North America, but over the course of every single year, all of the ticks that we receive, Lyme bacteria represents 65% of the pathogens that we find. It's that other 35% that I'm always trying to find places to talk to people and kind of get that on their radar so that also becomes part of a conversation with your doctor or whoever you're talking about following a tick bite, where we can get a very narrow focus on Lyme disease, and we can put in an order for a Lyme disease test. That is sort of, we could spend several sessions just talking about testing and what sort of the reliability of that test. But to me, it's more important. I can't personally change the Lyme disease test, but I can at least make sure that that's not the only question that we're thinking about. So this chart here shows us the infection rates just year over year across New England. So this is Connecticut, Rhode Island, Massachusetts, Vermont, New Hampshire, Maine, and that I've just pulled out the deer ticks from deer tick data from the years you see here, 2017 through 2024. And we show 5 different pathogens here. Up at the top by itself, that is Borrelia burgdorferi, causative agent of Lyme disease. And so clearly it's kind of at the top spot, and it changes a little bit. I'll get to that in a second. Down here, the next line, orange, is Babesia microti. Green, mostly overlapping with that, is Anaplasma bacteria. This lighter blue line is Borrelia miyamotoi, which can cause hard tick relapsing fever. And then the sort of purple line at the bottom, just skimming below 1% all the way, is Powassan virus. So these are all pathogens that are found in deer ticks. There are a few other pathogens they can carry. We don't find them terribly often, so they wouldn't really register on a chart like this. And it's really just a few per year. But as we look at these, there is a little bit of movement in the most common pathogens, Borrelia burgdorferi here, and then Babesia and Anaplasma. If we look at this chart, it looks like it's good news, and that if anything, the infection rates among ticks are declining year over year. I wouldn't go that far. I really say that I always describe the infection rates as surprisingly and kind of reassuringly steady year over year. We really don't see much of a change most years if we're looking at similar sample sizes. So here, when I'm looking at this, I am thinking about some other things that affect sampling. In 2020, all public health dollars went to something other than Lyme and tick-borne illness. And so a lot of funding that helped make testing more accessible to people, and that just focused on education and made sure that people knew that they were thinking about ticks, and that they knew about resources like the TickReport lab. That money disappeared. And so what happened is that 2017 through 2019, we were reliably testing between 5,000 and 6,000 ticks per year from New England alone. And then starting in 2020, 2021, that dropped to more like 2,500 to 3,000 ticks per year. So just different sampling, which can throw off percentages. There's probably a bit more that we could also find with life stages, where we probably were getting a slightly higher percentage of nymph stage ticks rather than adults. Nymphs have lower infection rates than adults. So yeah, but I always try to focus on the good news that infection rates in areas where we're getting a lot of ticks, they're not going up. There's lots of information on the internet that seems to be pointing in another direction. And again, I see that much more as a function of awareness and tick-borne illness getting the attention that it's taken a good while to finally get that sort of focus. So a lot of those studies I see as very positive. There are other infections that ticks can carry, especially other species. They tend to be lower. Deer ticks are always kind of public enemy number one, so I'm mostly focused on that here. And I want to run through a number of these slides. I'm happy to return to any of them and answer questions at the end, but going to sort of jump around with some topics because I'm really was trying to address a number of questions that attendees had sent in advance. So the next question was, what's the difference between Lyme and Babesia? So if we're talking about the pathogens, Borrelia burgdorferi for the most part, and then Babesia, there can be multiple species of Babesia parasites. Certainly the most common and the most associated with human illness in North America is Babesia microti. And there are a number of differences between the two of them. I am always trying to make sure that folks are aware of Babesia and how common it is in deer ticks, because many times when you have a tick bite, the first step may be to throw doxycycline or a number of other antibiotics against it. And in many cases, that can work fairly well. Doxycycline is a pretty decent multi-tool against many bacteria that ticks carry. It's there's certainly a lot of questions that we have about how effective it is in every case, how long that treatment needs to go on, but most of the pathogens that ticks carry are bacteria, and doxycycline in many cases is a really effective tool against all of them. Babesia is one of the big exceptions there because it's not a bacterium. It is a red blood cell parasite. It's very common. It is in most cases very treatable. The drugs you would need are available at any local pharmacy, but you have to know that it's a possibility, because doxycycline won't be effective against it. Instead, you would use an antiparasitic, usually supported by a different antibiotic, and one of the possible advantages with Babesia is that it is actually a blood-borne pathogen. So really all that Babesia wants to do is get into our red blood cells and eat those red blood cells and multiply. And that means that it stays where those red blood cells are, and it's in our bloodstream and potentially a little bit more detectable if we know to look for it. And it has an extra advantage where we don't always need to find it itself. We don't need to necessarily see the parasite, so we're not quite as affected by the just sheer number. You don't have to have your entire circulatory system overrun with Babesia parasites, because they damage and sort of shred red blood cells in a way that's fairly recognizable. So in many cases, the diagnosis is to take a drop of blood, put it on a microscope slide, and then you look for, you might see signs of the parasite itself, but you're probably in most cases going to be looking for these red blood cells that have been damaged in a particular way. And so you would see indirect evidence of that infection, but it lets you know that it's time to look deeper, and it gives you some options for treatment. And so number one, if folks are if they're bitten by a tick and immediately they're just hitting it with lots of doxycycline, again, that's pretty good with bacteria, but it's just not going to affect Babesia at all. So that just trying to overwhelm your system with antibiotics, I always worry that there can be a false sense of security there because Babesia wouldn't be affected. And it does seem that in many cases, Babesia infections are asymptomatic, at least in their early stages. And so it seems like an illness that it can be fairly common to live with, or an infection that can be fairly common to live with for a long time. And whether it's something that's recognized as an illness, whether you have a lot of kind of outwardly visible symptoms or not, that really seems to depend on the individual, and in some cases, it may be that those symptoms only appear very gradually over time. So again, it's something that can be a little bit hard to spot because it may be brushed off as, I don't know, as just the effects of aging or from something else that has happened. And so I suspect that it's probably an illness that's overlooked quite a bit. But the biggest difference I always try to push here is it's not a bacteria. It's not going to be affected by doxycycline. So with deer ticks in particular, it's always good to keep that on your radar and know that it requires a slightly different approach in treatment. Okay. Jumping topics again, just about PCR testing. PCR testing is something that most of us have heard about, and there's been a lot of news attention on it, especially since 2020 when PCR testing was part of many of our daily lives due to COVID. And what it does, it's a molecular method that just creates many, many copies of a specific segment of DNA if that DNA is present. So you basically provide very specific raw materials that will only mesh together with certain DNA targets. And so if that DNA is present, they get to connect and then divide into two, and then those two divide into four, and it will make potentially kind of limitless exponential growth until the raw materials that you've given it to make those copies runs out. It is extraordinarily sensitive. Just a few molecules, if we're thinking about Lyme, just a few spirochetes are potentially going to be detectable through PCR. And if the test isn't calibrated right, it might be too few spirochetes to actually cause infection. And unless you are, depending on the type of PCR you are doing, it's also possible that it's very easy to detect DNA from organisms that aren't alive anymore. So there could be dead Borrelia or sort of fragmented Borrelia in the sample, and so there is a quantitative portion. We can figure out, is this how much is present. You would have to take a different approach to try to figure out if that Borrelia is active and viable. And so it is something where we saw this debate in 2020, 2021, 2022 with COVID testing, where there was a lot of arguments about what the presence of, in that case, coronavirus RNA meant. It didn't always mean that the infection was still active. It's possible that the virus RNA was present and just kind of being worked out of the system. And so there was a real debate between what we really know about PCR and pressures to stop isolating. Those are totally separate here, but it's very sensitive. It needs to be calibrated very carefully. A lot of what we were doing a few years ago with COVID, we just didn't have enough knowledge to know at what point that infection ended. There's been a lot more research for a lot longer with ticks, and so we are in much better shape in terms of being able to interpret the results of PCR testing. But PCR, as it's used in tick testing, it's really asking more or less a yes or no question. And boy, can that question really be messed up if the researcher isn't being responsible and doesn't quite have the experience and the resources to just really pick that assay apart and make sure that they're getting it right. So if we think of a strand of DNA that, I don't know, I probably saw in Jurassic Park in an animated version there, but it's essentially like a ladder that is twisted, a spiraling sort of ladder. And that ladder is made up of little points of basically four essential bases, and they are just A, C, T, and G. So if we're thinking about Borrelia, the genome of Borrelia bacteria, so all of the DNA, all of the genetic information that goes into making Borrelia bacteria what it is, that is about 900,000 bases long. And it's 900,000 C's, G's, A's, and T's, just in a very, very specific combination. And so there's four possibilities in each. Think of it as a word that is 900,000 letters long, and it can only be written with A's, T's, C's, and G's. The challenge with this is that a huge portion of just any carbon-based life form, a huge portion of that DNA is shared. You often hear these things on sort of Discovery Channel type programming about how we share 99% of our DNA with animal X. We share an awful lot of our DNA with most things that we can see. And there are specific segments that set us apart. This shows the part of an assay that we used for Borrelia bacteria. So you look at Borrelia bacteria, that 900,000 letter word, and you try to find segments in that are where that series of letters is found in all Borrelia burgdorferi bacteria, and it's unique to that Borrelia burgdorferi bacteria. That takes a lot of work, and especially the unique part, because again, we share an awful lot of our DNA with many other animals out there, and if we aren't careful, it's very easy to find something that isn't unique, and you get a lot of false positives. So in that 900,000 letter word, we have found a segment of 200 letters that is unique to Borrelia, and we target it. Without getting into it too much, these are the beginning and end of that 200 letter segment there. And this is hard work. And a lot of times there are public databases that you can submit these sequences to, and they aren't necessarily, each sequence is not necessarily independently validated. So if you go to these databases that are just incredibly valuable, they're the foundation of so much work that's done in molecular biology, there are a lot of those sequences that are saying this sequence will let you identify organism X. And if you take them at face value and you're able to validate it yourself, you can cause a lot of problems there. And we see that in all sorts of areas, particularly in tick testing. We've seen that a number of times. I'll get to one of those examples in a little bit. So yeah, PCR testing, incredibly powerful. And so the technology itself is amazing, but you really need to know who is running it, because it's a powerful tool that is only as good as the person who's designing an assay with it. There's a question about PCR testing in humans, and I don't generally try to get into diagnosis and treatment too much. PCR is not always the first choice with diagnostic tests. Some of it might be related to cost, just because of scale. So there are a lot of other tests that are maybe cheaper. They might be more shelf stable. They're easier to have right in the office, that are a little bit easier, and they've been around longer, and so they have regulatory approval when a lot of PCR testing might not. That's a mix of both good and bad about PCR and about the way that our health systems are designed, but PCR is not always the best way to do it. There is that chance of a false positive, by where, again, kind of going to the COVID example, you can test positive for PCR well after the active infection is over. And so the challenge is figuring out what is the rate at which that positive rate would decline, so that we know that it's on its way out. You need a lot of time, you need a lot of human subjects to sort of calibrate that. That's hard to do, it's expensive, and if there's a test that makes some parties happy already, you don't necessarily, it can be difficult to reinvent the wheel. It's also, I always think of it, especially on the diagnostic side, especially with tick-borne illness, you're also limited by the type of sampling you're able to do, where a lot of the testing we do would be based on a blood sample, and you're really relying on a detectable amount of that pathogen or the antibody to be passing by in that vein at the very moment that the blood sample is taken. That's a pretty tall order, unless the infection is pretty advanced already. So in many cases, there are other ways where symptoms might be a better choice. With something like babesia, it might be easier to take that blood sample and look at it under the microscope instead of doing PCR. There's a number of options there. A lot is really just going to come down to what is accessible through your healthcare provider and your insurance. It's a pretty big area. There's a lot of things that are more complicated than I am able to talk about, certainly in one sitting. Testing of ticks and accuracy, again, if the assay is well designed, is much easier than using PCR on humans. And one of the big advantages is that we're going to grind up the entire tick, homogenize it, and then really anything that was anywhere in that tick, that pathogen DNA is going to be detectable. I sometimes do, folks come to the lab, or they email or they call or whatever, and they say, why can't we use this technology to diagnose people? And I said, well, in theory, we could, by taking a person and grinding them up and homogenizing them. And in theory, we could get a pretty definitive answer. It wouldn't do that person much good anymore. So it's not something that's particularly practical there. But one of the questions that we received was, is there a chance that a tick would not have detectable pathogen DNA if it had bit a person? So this is a question I get a lot, where if there is a tick that was infected and it bites you and it transmits Lyme bacteria or whatever pathogens it might have, is it possible that it just wouldn't be in the tick anymore? Nope. Very, very simple answer there. And you can think of it as just like passing a cold from one person to another. This isn't it's not kind of a demonic possession like we see in TV and movies, where you make contact and there's this single transfer from one to the other. If I have a cold and I am near someone else and unfortunately share my germs with them, I might be more advanced in it, so I might be closer to getting better, but we're both going to be having those pathogens active in us at the same time. And with ticks, it's going to be the same thing. Not all of the pathogen is going to move from one to the other. Partly, it's going to stay there. It's going to stay there for a while. Bear with me while this call goes to voicemail. I can't control the phone in the next room. But I will close the door. Hang on.
Thank you for your patience, everyone.
In those cases, some of the pathogen will stay in the tick because that's just its life cycle. It's going to stay there. But even if somehow all of the live bacteria was able to say, oh, the tick is feeding on a host, so let's all make our way into that new host, there would be remnants of bacteria that had sort of died. Bacteria doesn't, the same cell doesn't last all that long. So there's really not a likely practical way that a tick would rid itself of any of that infectious material. There are a few cases where ticks, by feeding on an animal that has blood that kills Borrelia, the tick can sort of get cured by that blood meal in a certain way. Most species of deer, their blood is Borreliacidal, which means it kills Borrelia. There is, in California, the western fence lizard is famous for having Borreliacidal blood, where ticks will, as larvae, they will feed on a rodent and they will become infected with Lyme bacteria. And then as a nymph, they will feed on this western fence lizard, and that lizard's blood will kill the Borrelia in that tick. And then when that tick becomes an adult, it's no longer infected. There's a few cases like that, but that's a totally different mechanism. That's where the blood that the tick is actually taking in is killing and destroying all of the Borrelia that is in that tick. But in terms of just a tick sort of spitting out all the pathogens that it contains and not being positive anymore, that really isn't a likely scenario. And then as sort of a last slide, hitting on a couple of topics here. Uh, tick testing, uh, tick-borne illness research is always a work in progress, and it's tough. You really can't responsibly say that this is what we know and what we will always know, and that we have learned everything that there is to know. Um, and there are sometimes it's important to know about just some of the natural sort of life cycle of scientific discoveries or scientific missteps. So an article will come out and there may be, it may claim a huge breakthrough or suggest that they might be on the verge of a big breakthrough, but that article by itself isn't the final word. It's the next several years or next several decades of people citing and responding to that article and trying to replicate the findings of that study where we really start to learn. Um, some things we just yeah, a researcher will miss something, or in technology may limit some of their identification. A couple years later, it becomes much easier to prove them wrong. It's not a statement on the value of the first person because they started the conversation, and they brought all of us forward. One of the most famous examples in my tiny part of the world is with Bartonella henselae. There was a publication several years ago that showed that said that it was based in parts of Europe that were finding very high infection rates of Bartonella henselae bacteria in the ticks that they collected. And a couple years later, an absolutely fantastic, kind, patient, thorough article said, yep, there was a fundamental flaw, and what they were finding was not Bartonella at all. It was just another organism that is present in the soil. It's all over the place. And because they had, there was a little bit of a problem with that section of that very long word made out of only 4 letters, it wasn't as specific and unique as they thought. And by repeating the experiment that way, they found out that Bartonella henselae was not present in any of those, and in the same regions, they couldn't find it in a single tick. Does that mean that Bartonella and ticks is a closed case? Absolutely not. Bartonella infection exists. Many people are infected with Bartonella. No one that I'm aware of has been able to draw that link with a tick bite itself. There are known vectors of Bartonella infection, and certainly what I think we can say is the same sort of very, very healthy behaviors that expose us to tick bites can also expose us to some of the insects that absolutely do bite animals and people and pass on Bartonella there. There may also just be new things that we're going to learn about Bartonella going forward. And in some areas, other species of Bartonella bacteria are carried by ticks. In parts of Central America, in certain amblyoma ticks, they can carry Bartonella quintana. There's a few dozen species of Bartonella. There's plenty to be learned, but with the best research that has been done with people very carefully looking over each other's shoulders and being part of that ongoing conversation. They, that connection to a tick bite hasn't been confirmed. But stay tuned. In 10 years, that may change quite a bit. The other real open question right now is really quite a number of species of Babesia are open questions, and one in particular is Babesia oticolii. However you want to pronounce it, you're welcome to do it. These Latin or fake Latin names, we're making it up as we go along, so I'm never a judge if you have a slightly different pronunciation. This is something that's getting a lot of attention. We have not added it to our testing at TickReport for a couple of reasons. Number one, it's not clear how often it is pathogenic, how often it can cause illness in humans. It is a species that exists. It's out there, and particularly in some hoofed animals, it does exist. So again, it's there. We certainly can't say it's impossible. We're at the beginning stages, I think, of figuring out if it is likely to cause disease in humans. And if you really dig into some of the early publications, the publications that I've read about it, there's a little bit of a challenge because it's based on those published sequences that aren't necessarily very vetted. And even the authors themselves have really approached it the right way and have said that, yeah, there is some question about how specific it is. And especially they're wondering if those molecular tests can tell the difference between Babesia oticolii and Babesia duncani, which we do test for and that we have tested for a number of years. So whether it's pathogenic or not is really important to me, because even as many times as I say that tick testing is not a diagnosis, we know that people take it very, very seriously and they may base treatment on those results. But even if they don't base treatment on it, seeing that positive result on a tick test is a source of anxiety. And just the position that we've taken is that until we know that this for sure, until we are much more confident that the result of that tick testing has a reasonable connection to what can happen down the line with the person who's bitten by it, it's just not a test that we're comfortable putting out there. It's the same reason that in many cases we avoid genus testing. So you'll see this in some cases where it would be Bartonella species, or maybe Bartonella abbreviated as Bartonella SPP. And that just means it's designed as a genus test that would detect every single species of Bartonella. And there's two issues there. Not every species of Bartonella are pathogenic to humans. So if you're just saying that some sort of Bartonella is here, again, we don't think that it's responsible to say that when we can't nail it down and say that it's one of the roughly 30%, I think, of species of Bartonella that can cause illness, causing a lot of extra anxiety and potentially unnecessary treatment when we can't identify it specifically. And Genus tests are very, very attractive because you can do a whole lot more with a single reaction. They're also very difficult to design. So by going back again to that Borrelia example, 900,000 letter word, we found 200 200 letters that are a series of 200 letters that we know is common and unique to all Lyme bacteria. If we are designing that to include Lyme bacteria and Borrelia miyamotoi and Borrelia lone stari, we have to start making some compromises. Sometimes you're sort of putting in wild cards into that, where instead of only using A's, T's, C's, and G's, you sort of allow for some of them to be anything, or say, all right, this one could be an A or a G. And the more ambiguity you introduce there in this search for sort of efficiency and simplicity, the more likely it is that you're going to sort of overcorrect, and now you're going to be detecting things that aren't even part of that genus, because you're giving up more of that unique sequence. Borrelia, enough work has been done over decades now that Borrelia genus tests are okay, and we use them. Other pathogens, I don't think we're quite there. So yeah, there's a few things there where genus test can both give you the sense that you're exposed to something infectious that might not be infectious, and it may get it wrong completely, just because you sort of built in enough ambiguity. This is something that has particularly bothered me with some studies that showed Lyme bacteria being carried in mosquitoes in parts of Europe. When you dig into those articles and you look at at the sequences, so those letter sequences that they have said this is what we're considering to be Borrelia, with the article that gets the most attention, close to 40% of those letters are wild cards. That's out of hand, and so it definitely raises questions. Again, we don't just say no, that can't be right. We also do some mosquito testing around here and mosquito surveillance. We've done quite a bit of mosquito surveillance in a neighboring town here that we know 50% of deer ticks carry Lyme, and we've tested all of those mosquitoes sort of on the side as a trust but verify approach to this question in an area where we know that Borrelia is hiding under every log. None of those mosquitoes had even the slightest hint of Borrelia bacteria. Doesn't mean it's impossible, but we feel pretty good that we have a really good sample area there, and then it's not quite there. That is, again, kind of jumping question to question. That is the end of my prepared remarks, but I'd love to hear if there are other questions and just open up the floor.
If you can either write in the chat, or if you'd like to raise your virtual hand, we're open to questions. And we have a question from Nellie. Can you describe the process of sending in a tick to TickReport?
Yeah, so when folks are bitten by a tick, carefully remove it. Tweezers is always the best move. Really, really sharp pointed tweezers that you can come in from the side, pull the tick up without squeezing as little of the tick as possible, but there's really nothing fancy about it. Our preferred container is just a zip top plastic bag from your kitchen, but people go to TickReport.com and order a test. In there, we ask for contact information, but we also ask for surveillance data about where do you think geographically you encountered this tick. And then that goes into our database that is publicly accessible, and it feeds into charts like this. This is a screen grab from our website, so anybody can go to TickReport.com, and up in the corner there's a statistics page link, and you can filter by zip code or state or tick species or pathogen, and you can, or over time, you can look for the last month, or you can look for the last year or the last 20 years. So that information that we ask about where do you think you picked up the tick geographically, and then where on your body did it bite, that all gets fed into that website anonymously, but is an important part of the ongoing research. At the end of that online form, you're just assigned a 6-digit sample ID, and if you can write that on the bag in marker, you can mail it to the lab or send it off. Mail it to the lab or drop it off if you're in the area, and then we guarantee all results of testing within 3 days of receiving the tick at the lab. So really very simple. You're not waiting for a kit to come out. Pretty simple online form. Get it in the mail. Ticks are surprisingly durable and remain testable for a long time. So there's no ice or any ice needed, no special padded envelopes. I do always say, whether you're looking to test a tick or not, if you just have a tick that you're going to hold on to for a little bit to see if symptoms emerge, there are many sources on the internet that tell you to put like a damp paper towel or a damp cotton ball or something into the bag. Please, 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 please don't. The idea behind that is to help with microscopy. It has no real effect on the DNA testing, and more often than not, if it is in that bag for more than 2 or 3 days, when it arrives in the lab, the whole bag is just covered with black mold. The water doesn't do anything. It doesn't do what people advising you on the internet to do think it accomplishes, and it really does make it much more difficult and mildly hazardous to handle. So just tick in a bag or whatever container is handy. There's no worries about ticks escaping. If you put it in a bag and it's crawling around, a couple of hours in your freezer will take care of that. And yeah, they are really quite durable. Our personal record for just to see if we could do it, we had a tick taken out of an old entomology collection at UMass. The tick was 114 years old. The DNA amplified incredibly well. And sometimes we'll receive something that the bag has been shredded in transit, a sorting machine with the postal service tore it apart or something. We can swab the inside of the bag with a Q-tip. I wouldn't rely on that for pathogen testing, but we can definitely figure out what species of tick it was. So those sort of trace elements are there. DNA testing seems close to magic sometimes in its sensitivity, but yeah, it really, tick testing and the ticks are much more durable than a lot of people think.
Thank you, Paul. I don't see any further questions from the audience. I do want to thank you for taking time out of your busy day to help us with our educational programming.
Sure thing.
And we hope to see you again soon.
Alrighty. Well, thank you very much. Thank you everybody for hanging in there. And if there are follow-up questions that you want to send along, please do. And ticks are going to be very, very active through at least July 4th. So make sure you're using all of your precautions for the next few weeks. And then always remember that there's a second fall season. Looking at this chart, beginning of, usually I say around October 15th, the deer ticks are out in force again, and they're usually active through Thanksgiving into December, depending on how cold it gets and when we get our first snow. I always think of that as a very dangerous time of year because I assume that since many tick-borne illnesses start with general flu-like symptoms, that if you start having something that seems like the flu in early November, well, I got that bug that's going around. And that's where I really worry that a tick-borne illness is going to be overlooked, not treated, and can become more serious at that point.
Words of wisdom. Thank you so much for coming this evening, and I thank our audience. And know that we will be posting this video on our YouTube channel, and we will be sharing it with all our participants this evening. Thank you and good night.